In 1961, blood from an indigenous Australian woman reached a laboratory and failed a test that should have identified at least one Rh antigen. Her red cells reacted with none of the available Rh antibodies, leaving investigators with a phenotype medicine had never documented before. 3 years later, the condition acquired a name that sounded almost impossible: Rh-null, blood with no detectable Rh antigens at all. The discovery was so unusual that it eventually earned the informal nickname golden blood, not because of color, but because compatible units are extraordinarily scarce. Yet the strangest part was not the transfusion value. It was what her blood revealed about how unfinished the human genome really is. Rh-null can arise when mutations disable the machinery that builds or displays one of the most important protein complexes on a red cell. Different families can reach the same rare phenotype through different genetic routes, including defects in RHAG or the Rh locus itself. In 2026, investigators reported another regulator type, Rh-null donor, carrying 2 different rHAG mutations, one of them newly described. The mutation had not bypassed inheritance. It had entered it, becoming another heritable change added to the enormous variation already circulating in humans. But rare does not automatically mean adaptive, and Rh-null is not evidence that evolution is intentionally designing a superior blood type. People with Rh-null can develop chronic hemolytic anemia, abnormal red cell shapes, shortened cell survival, and extreme transfusion difficulty. So the real evolutionary story runs in the opposite direction from the myth of a futuristic upgrade. New mutations continually create biological possibilities. Selection, drift, migration, reproduction, and chance decide what happens to them afterward. Some disappear, some persist quietly in families, some become harmful. And occasionally, environmental pressure makes an unusual blood variant advantageous. Blood groups provide one of the clearest places to watch that process, because their molecules sit directly where pathogens meet human cells. Malaria has repeatedly exploited red cell proteins as doors, while human mutations have repeatedly altered, weakened, or removed those same doors. ABO variants have survived across primate lineages for millions of years, suggesting that changing pathogens can preserve multiple competing forms. The Duffy-null phenotype became extremely common in much of sub-Saharan Africa because it strongly reduced susceptibility to Plasmodium vivax malaria. In Melanesia, a deletion affecting glycophorin C created Gerbic-negative red cells and interfered with another malaria invasion pathway. Those examples reveal what Rh-null alone cannot. Mutation creates novelty, but evolution becomes visible when that novelty changes survival and reproduction. Every child is born with new mutations that were absent from both parents, and every generation reshuffles older variants into new combinations. Rare blood types are therefore not blueprints of a planned transformation. They are snapshots of the raw material evolution is still working with. The first Rh-null sample made that hidden process visible because the Rh system is normally almost impossible to erase. The Rh blood group is not one antigen. It is a large and highly polymorphic system built mainly around proteins encoded by RhD and RhCe on chromosome 1, working with the related RhAg protein encoded on chromosome 6. Most people know only the D antigen because it determines whether a routine blood type is called positive or negative, but Rh includes dozens of recognized antigens, including C, E, and E. Their presence depends on closely related membrane proteins that sit within a larger red cell complex. Removing every detectable Rh antigen therefore requires a much more profound disruption than ordinary RhD negativity. The proteins are not decorative labels attached only for blood typing. RhD, RhCe, and RAG are embedded in the red cell membrane and interact with other membrane components. RAG is essential for normal expression of Rh proteins at the cell surface. When that assembly system collapses, the consequences extend beyond whether an antibody binds during a laboratory test. Rh-null cells can become stomatocytic or spherocytic, lose membrane stability, and survive for less time in circulation. The resulting Rh deficiency syndrome may produce mild or moderate chronic hemolytic anemia. That phenotype is the first clue that Rh-null is not an evolutionary improvement waiting to replace ordinary blood. The mutation can impose a real physiological cost. These roots matter because they demonstrate convergent phenotype rather than one ancient Rh-null founder spreading through humanity. Different mutations can independently produce blood cells that look serologically similar. In 2024, investigators described a novel homozygous frameshift deletion in RAG that prematurely truncated the protein and produced Rh-null in members of a family. Another 2026 report identified regulator type Rh-null caused by compound heterozygosity. 2 different RHAG variants inherited on separate copies of the gene. One was a newly characterized missense change. That person was a healthy blood donor, illustrating how dramatically the clinical expression can vary even when the serologic phenotype is extraordinary. The mechanism is completely traditional inheritance. Rh null does not somehow bypass Mendel. It usually appears when a person inherits damaging variants from both sides of a family, while relatives carrying only one altered copy may have normal or near normal blood. Consanguinity can increase the probability that the same rare recessive allele meets itself in a descendant. What makes the phenomenon evolutionary is not that inheritance has been escaped, but that inheritance is constantly supplied with new variation. DNA copying is extremely accurate, yet not perfect. Mutations arise in sperm and eggs during early development and across ancestral lineages. Recombination then shuffles existing variants, creating genetic combinations that have never existed before. Most new mutations do not redesign a visible human trait. Many occur in regions where they cause little measurable effect. Others are removed by purifying selection because they reduce survival or reproductive success. A rare mutation may remain confined to one family for generations simply because chance never carries it farther. Evolution contains enormous amounts of disappearance. Rh null may fit that pattern more closely than adaptation. The phenotype offers no established survival advantage in a modern environment. Its anemia can be disadvantageous, and transfusion becomes extraordinarily dangerous once an Rh null person forms antibodies against common Rh antigens. The rarity of the phenotype is not evidence that selection is pushing humanity toward it. At that point, compatible blood may have to come from another Rh null person. International rare donor networks exist precisely because some phenotypes are so uncommon that a suitable donor may be found in another country rather than another hospital. Frozen red cell inventories and family testing can become life-saving technology for a mutation that natural selection never promised to make convenient. The International Society of Blood Transfusion defines rare donors partly by absence of high-prevalence antigens or unusual combinations, occurring in fewer than roughly 1 in 1,000 donors. Some phenotypes are so scarce that fewer than 10 registered donors may exist globally. Rh null, Bombay, and several other antigen-negative states sit at the extreme end of that problem. That scarcity can create an illusion that a rare phenotype represents the next stage of human biology. Evolution does not work that way. New traits do not move from rare to common because they are unusual. Their frequencies change when carriers leave different numbers of surviving descendants, when populations migrate or mix, or when random genetic drift amplifies or loses an allele. To see actual adaptation in blood, the investigation has to move from Rh null to mutations whose frequencies reveal a selective battle. Few battles have reshaped the human genome more aggressively than malaria. Plasmodium parasites spend part of their life cycle inside red blood cells, turning membrane proteins into evolutionary pressure points. One of the clearest examples is the Duffy blood group. The Duffy antigen receptor for chemokines encoded by ACKR1 appears on red cells and historically served as a critical entry route for Plasmodium vivax. A regulatory mutation can silence Duffy expression specifically on red blood cells while leaving expression in other tissues. Across much of sub-Saharan Africa, that Duffy null allele rose to extraordinarily high frequency. For decades, P. vivax malaria was correspondingly rare in many populations with widespread Duffy negativity, although the parasite is now known to infect Duffy-negative people in some circumstances. The evolutionary signature remains powerful. A red cell surface change altered a parasite's access to its host. This is what positive selection can look like. A mutation does not need to make the body generally stronger. It can remove one molecular foothold used by a lethal pathogen. If carriers survive childhood more often and later have more descendants, the allele becomes more common. After enough generations, a local adaptation can look like a defining population difference. Laboratory work demonstrated that a major P. falciparum invasion ligand, EBA 140, interacts with glycophorin C, and that Gerbich-negative cells interfere with this pathway. The high frequency of the deletion in malaria-exposed Melanesian populations is consistent with strong historical selection. Once again, malaria has attacked red cells through multiple molecular routes, and humans carry multiple evolutionary responses. Sickle cell hemoglobin is the most famous, although it is technically a hemoglobin variant rather than a blood group antigen. Glycophorin structural variants, Duffy negativity, ABO effects on... ABO takes the story much deeper in time. The A and B forms are not recent human inventions. Related ABO variants occur in other primates, and genetic analyses indicate that major allelic classes have been maintained across lineages for millions of years. That persistence is difficult to explain through simple drift alone. One leading explanation is balancing selection. If different pathogens exploit different carbohydrate structures, no ABO phenotype remains universally optimal. A blood group that reduces one infection may increase susceptibility or severity in another. As pathogen environments shift, the relative advantage of A, B, or O can shift with them, preserving diversity rather than allowing one allele to eliminate the rest. That is evolution without a finish line. Natural selection does not search for a perfect blood type because the environment never presents one permanent problem. Pathogens evolve. Diet changes, populations move, climate shifts, medical technology changes survival. A variant that was costly in one setting may become neutral in another, while a previously harmless trait can acquire new consequences. The Bombay phenotype offers another lesson in how rare blood groups emerge. Ordinary A and B antigens require an H precursor built on red cells by the enzyme encoded by FUT1. In classical Bombay blood, non-functional FUT1 prevents H formation, so A and B cannot appear on the red cell surface even if the person's ABO genes encode functional A or B enzymes. Because Bombay individuals can make anti-H, ordinary type O, blood is not automatically safe for them. O cells still carry abundant H antigen. A Bombay recipient may therefore require blood from another Bombay donor, turning a mutation in a glycosyltransferase gene into a major transfusion challenge. In parts of India, the phenotype is far more frequent than in many European populations, reflecting population history and allele distribution. Rare variants such as Bombay or Rh null show the generative side of evolution. Mutations can create new phenotypes right now, but frequency alone cannot tell us why they exist. Founder effects can amplify an allele in a small or historically isolated population. Consanguinity can reveal recessive variants. Migration can transport them. Drift can raise or erase them without any adaptive advantage. That distinction is essential because evolution includes more than natural selection. Biological evolution is fundamentally change and heritable variation across generations. Mutation creates new alleles. Recombination reshuffles them. Selection changes frequencies according to reproductive consequences. Genetic drift changes them through chance. Gene flow moves them between populations. Humans remain subject to every one of those processes. Medicine has altered selection pressures, but it has not suspended inheritance. Caesarean delivery can change survival around childbirth. Antibiotics alter infectious mortality. Vaccination changes exposure. Assisted reproduction changes, which infertility barriers prevent descendants. Global travel creates unprecedented gene flow between populations that were once geographically separated. At the same time, culture creates new environments faster than genes can respond. Agricultural diets selected for adult lactase persistence in several pastoral populations. High-altitude living favored variants affecting oxygen physiology in Tibetans, Andeans, and Ethiopian highlanders through partly different genetic routes. Infectious diseases repeatedly remodeled immune and red cell genes. It also explains why blood group genes can carry signatures of both positive and balancing selection. A pathogen may strongly favor loss of one antigen in one region, while another pathogen preserves multiple forms elsewhere. Some antigen systems show striking geographic structure because historical exposure differed. Others remain diverse across vast evolutionary timescales. Modern medicine adds a new pressure that did not exist for most of human history: transfusion compatibility. An antigen-negative person who would once never encounter another person's red cells can now be exposed during surgery, trauma, childbirth, cancer treatment, or chronic transfusion. That exposure can create antibodies against antigens absent from their own cells. But this new medical environment is not known to be driving Rh null toward higher frequency. Transfusions generally occur after the genes determining blood type have already been inherited, and rare donor programs aim to reduce harm rather than alter reproduction. Calling Rh null a solution to modern environmental pressure therefore reverses the evidence. Rh null is better understood as a naturally occurring disruption that modern technology suddenly makes globally important. Before transfusion medicine, its major consequences were the intrinsic properties of the red cells and whatever effects those had on survival. Today, one original donor can provide a unit with extraordinary value to a genetically incompatible patient thousands of kilometers away. Technology has changed the meaning of the mutation without necessarily changing its evolutionary fitness. That distinction appears throughout human biology. Glasses transform the practical consequences of poor vision. Insulin transforms survival with diabetes. Surgery changes congenital disease outcomes. A genotype's medical importance can change dramatically, even if the underlying DNA remains identical. The 1961 Rh null discovery, therefore, sits at an unusual junction. It was not the birth of a new human lineage. The mutation, or mutations responsible, had already passed through earlier generations before anyone recognized the phenotype. What changed in 1961 was our ability to detect how far red cell variation could go. Family studies later showed that Rh null itself had more than one molecular explanation. The original Australian case was consistent with a regulator mechanism outside the Rh locus. Other families revealed amorph forms involving the Rh genes. Molecular genetics eventually identified Rhag as the key regulator gene for many cases, explaining why apparently normal RHD and RHCE genes could fail to produce surface Rh antigens. That discovery is evolutionary evidence in a precise sense. Biological systems contain dependencies. A mutation does not have to alter the antigen gene itself to erase an antigen. It can disable a partner required for assembly. Evolution therefore explores networks, not just individual genes. And similar phenotypes can appear through mutations at different points in the network. The 2026 compound heterozygous case makes that process visible in the genomic era. 2 altered RAG copies combined in one person and shut down normal Rh expression. One variant was newly described by the investigators. It was not proof of purposeful adaptation. It was proof that the catalog of human genetic variation is still incomplete because mutation and inheritance keep producing combinations medicine has not yet recorded. There's no reason to expect that catalog ever to become final. More than 8 billion people generate an enormous number of births, and each genome carries new sequence changes. Most will never become famous blood phenotypes. Some will vanish within one generation. Others will enter family lineages and remain hidden until 2 carriers have a child. If environmental conditions make one of those mutations beneficial, selection can begin acting immediately. Immediately in evolution does not mean that a population transforms in a decade. It means there is no biological barrier requiring humanity to wait for a new evolutionary era. Differences in survival and reproduction can change allele frequencies from the next generation onward. One reason blood group evolution is especially powerful is that the selective pressure can act at the first physical contact between host and pathogen. A mutation that changes a membrane protein may alter invasion before fever, antibodies, or inflammation ever begin. That gives red cell genes a direct route from molecular variation to survival. When childhood mortality from infection is high, even a modest difference in susceptibility can translate into a large evolutionary effect across many generations. Yet modern selection can run in subtler directions. A variant does not need to save someone from a spectacular epidemic to evolve. If it changes fertility, age at reproduction, pregnancy survival, childhood survival, or the number of descendants by a small amount, allele frequencies can move over time. Researchers can detect such pressures using pedigrees, ancient DNA, biobank-scale genomes, and comparisons between historical and modern populations. Ancient DNA has made this process almost visible frame by frame. Instead of inferring every change from living people, geneticists can now sample genomes from individuals separated by centuries or millennia and watch certain alleles rise, fall, or move geographically. Blood-related variants can therefore be placed against changing environments rather than treated as timeless labels. The human genome becomes a historical record whose pages were written by disease, migration, diet, population crashes, and reproduction. Modern examples are often difficult to measure because medicine, migration, contraception, social inequality, cultural preferences, and changing a family size all influence reproductive outcomes. Selection can also act on thousands of variants with extremely small effects rather than one dramatic mutation. Evolution in a large technological society is harder to narrate, not absent. Rare blood types expose that complexity because they separate mutation from adaptation so sharply. Rh null is dramatic, but has no demonstrated adaptive spread. Duffy negativity is common and carries a strong historical malaria selection signal. Gerbich negativity is geographically concentrated and tied to a parasite invasion pathway. ABO diversity may persist because no one antigen pattern wins every infectious contest. The blood cell becomes a record of those decisions. An Rh null cell reveals a disrupted membrane complex. A Duffy null cell reveals the absence of a parasite receptor. A Gerbich negative cell reveals altered glycophorin architecture. An O cell reveals a different carbohydrate surface from A or B. None is simply more evolved than the others. That phrase, more evolved, is itself biologically misleading. Every living human belongs to a lineage that has been evolving for the same amount of time since our shared ancestors. But rare mutation is not automatically advanced, and a common allele is not primitive. Evolution describes descent with modification, not a ladder ascending toward biological perfection. The future will therefore not consist of Rh null people gradually replacing everyone else unless some extraordinary and currently unknown selective force made that phenotype consistently increase reproductive success. There is no evidence for such a process. Its intrinsic red cell abnormalities make that scenario particularly difficult to justify. What Rh null does prove is more basic and harder to sensationalize incorrectly. The machinery of heredity is still generating variants capable of altering fundamental human traits. What malaria-associated blood groups prove is the second half. When those variants interact with powerful environmental pressures, their frequencies can change dramatically across populations. Our species is still subject to both processes today. New pathogens can alter selective landscapes. Existing pathogens can change host range. Climate can move disease vectors. Diet and pollution alter physiology. Reproductive technology changes, which conditions prevent parenthood. Migration mixes alleles that evolved under different regional histories. Some of these pressures may eventually leave detectable genomic signatures. Others will disappear before genes respond. Cultural adaptation is often faster than biological adaptation. Humans can invent sanitation, vaccines, air conditioning, or new foods in a fraction of the generations required for strong allele frequency change. Technology does not stop evolution. It becomes part of the environment in which evolution operates. R.H. Knoll revealed that the Rh complex contributes to red cell membrane integrity. Bombay blood revealed the dependence of ABO expression on H antigen synthesis. Duffy Knoll helped expose a parasite's dependence on a host receptor. Gerbich negativity helped map another route used during malaria invasion. Human variation becomes a tool for understanding human biology. And sometimes that understanding arrives because a transfusion fails. A blood bank expects compatible units based on ABO and RHD, but cross-matches unexpectedly react. Specialists test more antigens, sequence genes, trace relatives, and discover a phenotype absent from routine panels. A medical problem becomes another entry in the map of human genetic diversity. The map keeps expanding because our species was never genetically uniform. Rare donor registries are practical databases built around that fact. Their purpose is not only to catalog exotic phenotypes, but to prevent a patient from dying because the blood most people can receive contains an antigen that one person's immune system cannot tolerate.